All right, let's dive right in. Today, we're talking about how brand new cannabis strains get made. And I don't mean by accident. We're talking about some serious science. It's a journey, really, that takes us from just two parent plants all the way to one totally unique, stable creation. So let's get into it.
You know, when you find a strain with that perfect flavor or effect, have you ever just stopped and wondered, how did they even do that? Well, I can tell you it's definitely not just random luck. Nope, it's a super deliberate scientific process, all designed to give you that specific experience.
Okay, so to really get how this all works, we've got to start with a fundamental concept: the difference between what a plant could be and what it actually becomes. I love this analogy. It's like the difference between an architect's blueprint and the actual finished building.
So let's break that down. You've got two key terms here. First, the genotype. Think of this as the plant's DNA. It's unchangeable genetic code inherited from its parents. This is the absolute potential, what the plant could be. But then you have the phenotype, and that's everything you can actually see, smell, and feel, the final result. It's the plant structure, its aroma, its potency, the whole shebang.
And this is where it gets really interesting. The final plant, the phenotype, isn't just a copy-paste of its genes. It's more like an equation: the genotype plus the environment it grows in. Things like light, water, even the nutrients in the soil, they all have a massive impact on which genes get switched on or off. That's exactly why you can take two clones, genetically identical, grow them in different spots, and end up with two pretty different plants. All right, so now that we've got that foundation, let's get into the action. The very first thing a breeder has to do is pick the parents. And trust me, this is no random choice. It's more like strategic genetic matchmaking, and it really sets the tone for the entire project. Every breeding project starts with a goal, right? So the breeder makes a wish list. What are they looking for? Maybe it's a specific chemical profile, super high THC, or that classic citrusy terpene smell. Or maybe they need practical traits, like a plant that's tough against mold or gives a huge yield. And maybe the most important thing: are the parents stable? You want them to actually pass on those good genes reliably. And a lot of the time, this means borrowing a trait from one plant to give to another. A perfect example of this is the auto-flowering trait. You see, breeders took this wild, low-potency cannabis plant called ruderalis, which honestly nobody really cared about for its effects, and they crossed it with these amazing high THC strains. Why? All for one reason: to steal that one awesome gene that lets it flower automatically. No light cycle change needed. Just brilliant, really. Okay, the perfect parents have been selected. Now it's time for the magic to happen: the first cross. This creates what we call the F1 hybrid generation. And these plants, well, they're pretty special. So what is an F1 hybrid? Simple. It's the first filial generation, which is just a fancy way of saying it's the direct kid of those two parent plants we picked, P1 and P2. And this has to happen in a totally controlled space, like a sealed tent. You've got to make sure only the pollen from the male you want gets to the female you want. No accidents allowed. Now, because we started with two stable parents, this F1 generation is amazingly consistent. Like, almost every single seed will grow into a plant that's a spitting image of its siblings. They also tend to have something called hybrid vigor, which means they're often stronger and healthier than either of their parents. And for a commercial grower, that kind of predictability is pure gold. But here's the twist. The F1s are great, they're consistent, but they're not where you find the really new stuff. For that, you have to go to the next generation. And this is where we start playing the genetic lottery. Just look at this. It tells the whole story. When you cross two of those stable F1 plants together, the next generation, the F2s, is a completely different ballgame. The genetic variability just explodes. The F1s were all nice and uniform, but the F2s, they're all over the map. You get a massive range of different traits popping up. Now you might be thinking, wow, that sounds like a mess. But for a breeder, this chaos isn't a problem. It's the whole point. It's in this wild, unpredictable F2 generation that they have the best chance of finding a total unicorn, a plant with a rare, amazing combination of genes that nobody has ever seen before. But it's not total chaos. Some things are still predictable thanks to basic genetics. Take the main cannabinoid type.
If you cross two F1 parents that carry genes for both THC and CBD, their F2 kids will follow a perfect 1-to-1 ratio. You'll get 1 part pure THC, 2 parts with a mix of THC and CBD, and 1 part pure CBD. It's like a little island of predictable science in a sea of genetic randomness.
And this really gets at a crucial point, the difference between the type of cannabinoid and the amount. See, picking a plant that's going to be THC dominant, that's pretty straightforward. It's controlled by just one gene. But trying to breed for a super high THC percentage, like pushing from 25% to 30%, whoa, that is way, way harder. It's a complex trait controlled by a bunch of genes all working together, which makes it super unpredictable.
Okay, so let's say our breeder has done it. They've gone through hundreds, maybe thousands of these F2 plants and found it, the one, the perfect plant. So now what? Well, now comes the hard part. Taking that 1 in a million find and making its amazing traits stable so you can get them again and again.
And the main tool they use to do this is a technique called back-crossing. The concept is actually pretty simple, but it's super powerful. You take that winning F2 plant and you cross it back with one of its original parents, specifically the parent that has the traits you're trying to reinforce.
So here's how it works, step by step. You take your keeper plant from the F2 generation. You cross it back to that original parent. Then, from those kids, you pick the best one and, you guessed it, cross it back to the parent again. You keep doing this generation after generation. Each time you're strengthening the genetics you want. It's kind of like you're using the parent as a template to copy and paste the good traits until the whole strain becomes stable and reliable.
Now, as you can imagine, this whole cycle of growing, selecting, and back-crossing, it can take a long time. We're talking years. But today, technology has given breeders an amazing shortcut, a way to move into the fast lane and work with a level of speed and precision that was just impossible before.
Yeah, the biggest enemy of old-school breeding is just time. Think about it. You have to grow every single plant all the way to maturity just to see what you've got. You're waiting months just to look at the flowers, and then you still have to send samples to a lab to find out what the cannabinoids and terpenes are. It's slow, it's expensive, and it takes up a ton of space. But then, along comes marker-assisted selection, or MAS, and this is a total game changer. Basically, it lets a breeder take a tiny little piece of a leaf from a seedling, just a baby plant, and run a DNA test. This test looks for specific genetic markers that are tied to the traits they're looking for. It's like having X-ray vision to see the plant's genetic code right from the very beginning. And the advantages are huge. We're talking about taking a plant that's only 2 weeks old and, with one test, finding out if it's male, if it's going to be a THC or CBD producer, if it has that autoflower gene, or even if it's resistant to certain diseases. It just completely short-circuits the old process. What used to take years of guesswork now becomes a fast, precise, scientific operation. So when you boil it all down, that's what this is. It's this incredible journey. You start with 2 parents, you dive into a massive genetic lottery, and you come out the other side with one stable, totally unique strain that you can count on every single time. And all this really leads to a big question about the future, doesn't it? With technology this precise, are we on the verge of creating truly designer strains? I mean, could a breeder one day just custom build a plant for a specific effect, just like ordering a pizza? The science says we are getting closer and closer. Pretty wild to think about.